with you guys. So another very famous term which we should know very accurately is the difference between the active immunity and the passive immunity. The active immunity is that when somebody is got infected with a pathogen, if somebody is got infected with a pathogen, its innate immune system is not strong enough or not able enough to deal with that pathogen. It triggers the adaptive immune system. The adaptive immune system understands the problem, sense the problem, communicate that problem to the T cells, cell-mediated immunity or adaptive immunity. And then the T cells got activated and activate the beta cells. T cells start killing the pathogen. Beta cells start producing the antibodies, and we can get rid of that pathogen. And in the meanwhile, certain memory cells are produced to store the information of that activity. That procedure is called as an active immunity. When our immune system contribute in fighting against a disease fully, using all its resources. Because that activity, if completed, using all the components of the immune system, that will end up in creating or in the creation of memory cells. And those memory cells are the precious information we are keeping in our body that can be used later on to fight against the same pathogen with in a lesser amount of time and in a stronger way, those memory cells. This is called active immunity. Now, what if the other topic, what if we inject the antibodies of a pathogen? If some person is fighting against a pathogen, if some person is fighting against the virus or bacteria, its innate immune system failed to respond against the virus. Its adaptive immune system is also failed to respond against that virus or bacteria. He failed to sense the problem, or he fails to communicate, or he fails to find or create an appropriate response or appropriate weapon. That person will got sick. He has no answer against the problem, like against the SARS-CoV-2, and the person developed the disease which we call COVID-19. Now, in that case, we look for the people who have already fought against the COVID-19, who have already fought against the SARS-CoV-2. Their body, their cellular adaptive immune system, when they found the virus, they learned about it, they communicate that information efficiently, they activated the T cells comfortably, and the T cells and the beta cells together with the help of antibodies and other cellular actions, they're able to destroy the virus efficiently. And also they're able to produce the memory cells. They contain the antibodies. Antibodies are present in the plasma cells, and those antibodies are the antibodies which are capable of destroying or killing the SARS-CoV-2, and that they have achieved through their active immune system or active immunity. If we borrow those antibodies, if we borrow the plasma, so not blood plasma, if we borrow the plasma from those people who fought against the virus successfully, their plasma contains those antibodies. If we get those antibodies and inject those antibodies into the person who is trying or who is fighting against the SARS-CoV-2, that antibodies should be able to interact with the virus and should be able to kill that virus. And this treatment is called as the plasma therapy. There are a lot of sensitivities associated with that disease. One of the sensitivities is that what if the other things are not matched between the two people, blood grouping. So we need to find the person who has the right blood group as well. And even when the blood group is matched, cross match karte hain, there might be certain other thing which might not be matched. Another problem, maybe the antibodies are not enough and the virus is locked in the recipient's body. Maybe it is not enough to kill that person, kill that SARS-CoV-2. A lot of complications also. And one of the doomsday complication is that, which we used to observe in case of cancers, is that you borrow the antibodies, inject it into the person who is fighting against SARS-CoV-2, but the person is fighting against a little bit newer version of the SARS-CoV-2. SARS-CoV-2 got mutated. So whether the antibodies are fine, is perfect, strong, those antibodies will not going to kill the SARS-CoV-2, which is now changed a bit. That's very, very dangerous. We don't want to know that SARS-CoV-2 is actually mutated. In that case, whatever the immunity we are developing, the herd immunity or the other sort of immunity, the people who have developed immunity, all those immunities got failed. With the second infection की probability है, the chance of second infection is only possible, number one, either during the first time we fail to activate our active immune system perfectly, memory cell नहीं बना पाए, antibodies are not enough, somehow we avoid the problem, but we fail to use our immune system in a perfect way. एक वजह ये हो सकती है. The second reason might be that, which is very dangerous, the virus is changing. So the person just received the SARS-CoV-2 once again in a very new way, got sick. That's very sad. That's very dangerous as well. Shabash ji. Shabash, good question. नाम क्या है? Aisha. This question belongs to this slide. That's a good question. कि vaccination में क्या actually क्या होता है? Vaccination passive immunity या active immunity करती है? क्या करती है vaccination? जब हम vaccination देते हैं तो हम किस चीज को trigger करते हैं? Active immunity को. Vaccinations में we are giving the pathogens. अगर देखो अगर हम vaccination में antibodies देंगे, we will kill the problem, but we will not going to activate the person's own immune system. But if we give the person what a pathogen, वो अलग बात है कि वो pathogen जरा lazy होता है. The pathogen is impotent, not capable of producing a disease. SARS-CoV-2 not capable of producing a disease is a vaccine. If we have a perfect SARS-CoV-2 structure but not capable of producing a disease is a vaccine, and that vaccine is going to trigger what? Trigger our rangers, macrophages, dendritic cells, then the T cells and the beta cells. The things will complete and eventually produce the antibodies as well as the memory cells. The polio vaccination में हम polio virus देते हैं ताकि kid develop what? The memory cells against that polio virus. So when during the lifetime when actually the virus came to the kid's body, the body is already ready and prepared. Will not going to take the precious time. The initial precious time is very important, because if you lose the battle, you will develop a disease. Developing a disease is not that dangerous if the disease is not taking your life, if the disease is not causing an irreversible damage. In case of polio virus, it is an irreversible damage. In case of SARS-CoV-2, it's a life-threatening situation. I mean, still 3% is quite scary. 3% is quite scary. I told you on the other day, that means a million deaths in millions. This is still a scary thing to imagine. Okay? So that is how the active and passive immunity work. I have explained that slide. Just see and you will came to know that we know all this wonderful. If I ask a question that in case of an active immunity, whether the cells will be able to develop memory, the answer is yes. Whether the cells will be able to develop the memory, the answer is yes. The cells will be able to do it. If I ask you, in case of passive immunity, if we inject the borrowed antibodies to the person's body, the person will fight against the trouble, the answer is yes. The person will develop the memory, the answer is no. If the same trouble come again to that person, the person needs to fight once again. That's a difference. But passive immunity is a way to exist, it's a way to survive, is a way to fight against the disease. If the person's immune system is not responding, or it's actually not responding is not a good word. If a person's immune system is failed to find the right answer. And I will show you in the future that it is all about probability at the end of the day. So whether you are a very macho person, you are completely healthy and strong, it is still possible you might not be able to find the appropriate combination. That is why, I mean, the people who are very strong and healthy, they can got sick because of things we are not. So that is entirely possible. So there are certain important definitions in that slide you should know, and this slide already explained. Just see and feel that you know the things present on that slide. I will share that book as well during this week to you people, so you can go through and you can feel that you know the things already which you are reading. That's very beautiful feeling. अगर आपको textbook पढ़ रहे हो अच्छे किस्म की, and you are feeling, oh, I know what that means, you don't need to work very hard in order to understand the things, then I will be happy at the end of the day that we have done something reasonably fine. Okay, just see the picture and think about that. When we have the primary we have an antigen. You can consider it as a bad thing. You can consider it as a pathogen. Anti-X, is a pathogen named X, a bacteria named X-Men. So if a pathogen enters into our body, our cells, naive T cells or beta cells, they will start fighting against the bad thing. They will try to learn about the antigen X. They will try to respond it. They will take some time, and after that, they will produce the antibodies, and those antibodies are good enough to finish and to kill the disease. So serum antibody level increases, and those antibodies will going to destroy the bad things, and then the antibody level will decrease. And we develop memory cells. Can you see? We develop the memory cells. We don't need to keep all the cells. We just need to keep these few memory cells in our body. That is good enough. ये बचत है ना? That's good enough. What about when another new antigen came? Why? At the same time, if the same antigen came as well, if we are infected with the same bad thing means infected with the X and with a new thing called as the Y. What will happen in that case? For our body, the Y is entirely a new thing. So we learn about the Y, we increase the serum antibody level against the Y, and then we develop the memory cells, and those memory cells are the memory cells against the Y in the same way. But how our body respond to the X? The X was already there, so our body contains the memory cells. Those will be used to create a very strong response in a very short period of time, and that will create a lot of antibodies in a very short period of time that can kill the things and then keep the beta cells for the X. So the X can be done very easily, strongly, because it is like the second exposure, but the Y will be taken as the first one. So no matter how many viruses and bacteria we are receiving today or at any moment, those will be considered and dealt in a different ways. It's a very strong and very efficient job for our immune system to think whether it was a new, it was a bad, we found it earlier, or we don't found it earlier, blah, blah, blah things.